Unit 7 Genetics and Evolution. The work of Mendel and others who followed him gave us an idea of inheritance patterns. However, the nature of those factors which determine the phenotype was not very clear. As these factors represent the genetic basis of inheritance, understanding the structure of genetic material and the structural basis of genotype and phenotype conversion became the focus attention in biology for the next century. The entire body of molecular biology was a consequent development with a major contribution from Watson, Crick, Nuremberg, Khorana, Kornberg's father and son, Benger, Monod, Brenner, etc. A parallel problem being tackled was the mechanism of evolution. Awareness in the areas of molecular genetics, structural biology, and bioinformatics have enriched our understanding of the molecular basis of evolution. In this unit, the structure and function of DNA and the story and theory of evolution have been examined and explained. James Davey Watson was born in Chicago on 6 April 1928. In 1947, he received BSc degree in zoology. During these years, his interest in bird watching had matured into a serious desire to learn genetics. This became possible when he received a fellowship for graduate study in zoology at Indiana University, Bloomington, where he received his PhD degree in 1950 on a study of the effect of hard X-rays on bacteriophage multiplication. He met Crick and discovered their common interest in solving the DNA structure. Their first serious effort was unsatisfactory. Their second effort, based upon more experimental evidence and better appreciation of the nucleic acid literature, resulted early in March 1953 in the proposal of the complementary double helical configuration. Francis Harry Compton Crick was born on 8 June 1916 at Northampton, England. He studied physics at University College, London and obtained a BSc in 1937. He completed PhD in 1954 on a thesis entitled X-ray diffraction, polypeptides and proteins. A critical influence in Crick's career was his friendship with J.D. Watson, then a young man of 23, leading in 1953 to the proposal of the double helical structure for DNA and the replication scheme. Crick was made an FRS in 1959. The honors to Watson and Crick include the John Collins Warren Prize of the Massachusetts General Hospital in 1959, the Lasker Award in 1960, the Research Corporation Prize in 1962, and above all, the Nobel Prize in 1962. Chapter 4 Principles of Inheritance and Variation. Have you ever wondered why an elephant always gives birth only to a baby elephant and not some other animals? Or why a mango seed forms only a mango plant and not any other plant? Given that they do, are the offspring identical to their parents or do they show differences in some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other or sometimes even so different? These and several related questions are dealt with scientifically in a branch of biology known as genetics. This subject deals with the inheritance as well as the variation of characters from parents to offspring. Inheritance is the process by which characters are passed on from parent to progeny. It is the basis of heredity. Variation is the degree by which progeny differ from their parents. Humans knew from as early as 8000 to 1000 BC that one of the causes of variation was hidden in sexual reproduction. They exploited the variation that were naturally present in the wild population of plants and animals to selectively breed and select for organisms that possessed desirable characters. For example, through artificial selection and domestication from ancestral wild cows, we have a well-known Indian breeds. Example, Sahiwal cows in Punjab. We must, however, recognize that though our ancestors knew about the inheritance of characters and variation, they had very little idea about the scientific basis of these phenomena. 4.1 Mendel's Law of Inheritance. It was during the mid 19th century that headway was made in the understanding of inheritance. Gregor Mendel conducted hybridization experiments on garden peas for 7 years, that is from 1856 to 1863, and proposed the laws of inheritance in living organisms. During Mendel's investigation into inheritance pattern, it was for the first time that statistical analysis and mathematical logic were applied to problems in biology. His experiments had a large sampling size which gave greater credibility to the data that he collected. Also, the confirmation of his inferences from experiments on successive generations of his test plants proved that his results pointed to general rules of inheritance rather than being unsubstantiated ideas. Mendel investigated characters in the garden pea plant that were manifested as two opposing traits. Example, tall or dwarf plants, yellow or green seeds. This allowed him to set up a basic framework of rules governing inheritance, which was expanded on by later scientists to account for all the diverse natural observation and the complexity inherent in them. Mendel conducted such artificial pollination or cross pollination experiments using several true breeding pea lines. A true breeding line is one that, having undergone continuous self pollination, shows the stable trait inheritance and expression for several generations. Mendel selected 14 true breeding pea plant varieties as pairs which were similar except for one character with contrasting traits. Some of the contrasting traits selected were smooth or wrinkled seeds, yellow or green seeds, inflated, that is full or constricted, green or yellow pods, and tall or dwarf plants. Figure 4.1, Table 4.1. Table 4.1, Contrasting traits studied by Mendel in P. Characters: number 1, stem height. Contrasting traits were tall or dwarf. Characters: number 2, flower color. Contrasting traits were violet or white. Characters: number 3, flower position. Contrasting traits were axial or terminal. Characters: number 4, pod shape. Contrasting traits were inflated or constricted. Characters: number 5, pod color. Contrasting traits were green or yellow. Characters: number 6, seed shape. Contrasting traits were round or wrinkled. Characters: number 7, seed color. Contrasting traits were yellow or green. 4.2 Inheritance of one gene. Let us take the example of one such hybridization experiment carried out by Mendel, where he crossed tall and dwarf pea plants to study the inheritance of one gene, figure 4.2. He collected the seeds produced as a result of this cross and grew them to generate plants of the first hybrid generation. This generation is also called the filial progeny or the F1. Mendel observed that all the F1 progeny plants were tall like one of its parents. None were dwarf, figure 4.3. He made similar observation for the other pairs of traits. He found that the F1 always resembled either one of the parents and that the trait of the other parent was not seen in them. Mendel then self-pollinated the tall F1 plants, and to his surprise he found that in the filial 2 generation some of the offsprings were dwarf. The character that was not seen in the F1 generation was now expressed. The proportion of plants that were dwarf were 1 by 4th of the F2 plants, while 3/4 of the F2 plants were tall. The tall and dwarf traits were identical to their parental type and did not show any blending. That is, all the offsprings were either tall or dwarf, none were of in between height, figure 4.3. Similar results were obtained with the other traits that he studied. Only one of the parental traits was expressed in the F1 generation, while at the F2 stage both the traits were expressed in the proportion 3:1. The contrasting traits did not show any blending at either F1 or F2 stage. Based on these observations, Mendel proposed that something was being stably passed down unchanged from parent to offspring through the gametes over successive generations. He called these things as factors. Now we call them as genes. Genes, therefore, are the units of inheritance. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as alleles. That is, they are slightly different forms of the same gene. If we use alphabetical symbols for each gene, then the capital letter is used for the trait expressed at the F1 stage and the small alphabet for the other trait. For example, in case of the character of height, capital T is used for the tall trait and small t for the dwarf, and capital T and small t are alleles of each other. Hence, in plants, the pair of alleles for height would be capital T, capital T, capital T small t, or small t small t. Mendel also proposed that in a true breeding, tall or dwarf pea variety, the allelic pair of genes for height are identical or homozygous capital T and small t respectively. Capital T and small t are called the genotype of the plant, while the descriptive terms tall and dwarf are the phenotype. What then would be the phenotype of a plant that had a genotype capital T, small t? As Mendel found the phenotype of the F1 heterozygote capital T small t to be exactly like the capital T parent in appearance, he proposed that in a pair of dissimilar factors, one dominates the other as in the F1 and hence is called the dominant factor while the other factor is recessive. In this case, capital T for tallness is dominant over small t for dwarfness, that is recessive. He observed identical behavior for all the other characters or trait pairs that he studied. It is convenient and logical to use the capital and lower case of an alphabet symbol to remember this concept of dominance and recessiveness. Do not use capital T for tall and small d for dwarf because you will find it difficult to remember whether capital T and small d are alleles of the same gene or character or not. Alleles can be similar as in the case of homozygotes capital T and small t, or can be dissimilar as in the case of heterozygote capital T small t. Since the capital T small t plant is heterozygous for genes controlling one character, that is height, it is a monohybrid, and the cross between capital T and small t is a monohybrid cross. From the observation that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and dwarf plant produce gametes by the process of meiosis, the alleles of the parental pair separate or segregate from each other and only one allele is transmitted to a gamete. This segregation of allele is a random process and so there is a 50% chance of a gamete containing either allele. As has been verified by the results of the crossings, in this way the gametes of the tall capital T plants have the allele capital T and the gametes of the dwarf small t plants have the allele small t. During fertilization, the two alleles capital T from one parent say through the pollen and small t from the other parent, then through the egg are united to produce zygote that have one capital T allele and one small t allele. In other words, the hybrids have capital T small t. Since these hybrids contain alleles which express contrasting traits, the plants are heterozygous. The production of gametes by the parents, the formation of the zygote, the F1 and F2 plants can be understood from a diagram called Punnett square as shown in figure 4.4. It was developed by a British genetist Reginald C. Punnett. It is a graphical representation to calculate the probability of all possible genotypes of offsprings in a genetic cross. The possible gametes are written on two sides, usually the top row and left column. All possible combinations are represented in boxes below in the square, which generates a square output form. The Punnett square shows the parental tall capital T male and dwarf small t female plants, the gametes produced by them and the F1 capital T small t progeny, the F1 plants of genotype capital T small t are self-pollinated. The symbols of this and this are used to denote the female that is eggs, and males, that is pollen of the F1 generation respectively. The F1 plant of the genotype T small t, when self-pollinated, produces gametes of the genotype T and small t in equal proportion. When fertilization takes place, the pollen grains of genotype T have a 50% chance to pollinate eggs of the genotype T as well as of genotype small t. Also, pollen grains of genotype small t have a 50% chance of pollinating eggs of genotype T as well as of genotype small t. As a result of random fertilization, the resultant zygotes can be of the genotypes T small t, or small t small t. From the Punnett square, it is easily seen that 1/4 of the random fertilization leads to T, 1 by 2 lead to T small t, and 1/4 to small t. Though the F1 have a genotype of T small t, but the phenotypic character seen is tall. At F2, 3/4 of the plants are tall, where some of them are T while others are T small t. Externally, it is not possible to distinguish between the plants with the genotypes T and T small t. Hence, within the genotypic pair of T small t, only one character T tall is expressed. Hence, the character T or tall is said to dominate over the other allele small t or dwarf character. It is thus due to this dominance of one character over the other that all the F1 are tall, though the genotype is T small t. And in the F2, 3/4 of the plants are tall, though genotypically 1 by 2 are T small t and only 1/4 are T. This leads to a phenotypic ratio of 3/4 tall, which is 1/4 T plus 1 by 2 T small t, and 1/4 small t. That is a ratio of 3:1, but a genotypic ratio of 1:2:1. The 1 by 4 is to 1 by 2 is to 1 by 4 ratio of T is to T small t is to small t is a mathematically condensed to the form of the binomial expression ax plus by whole square that has the gametes bearing genes T or small t in an equal frequency of 1 by 2. The expression is expanded as given below. 1 by 2 T plus 1 by 2 small t to the power whole square is equal to 1 by 2 T plus 1 by 2 small t into 1 by 2 T plus 1 by 2 small t is equal to 1 by 4 T plus 1 by 2 T small t plus 1 by 4 small t. Mendel self-pollinated the F2 plants and found that dwarf F2 plants continued to generate dwarf plants in F3 and F4 generations. He concluded that the genotype of the dwarf was homozygous small t. What do you think he would have got had he self-pollinated a tall F2 plant? From the preceding paragraph, it is clear that though the genotypic ratio can be calculated using mathematical probability by simply looking at the phenotype of a dominant trait, it is not possible to know the genotypic composition. That is, for example, whether a tall plant from F1 or F2 has T or T small t cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from F2 with a dwarf plant. This is called a test cross. In a typical test cross, an organism, that is P plants here, showing a dominant phenotype and whose genotype is to be determined is crossed with the recessive parent instead of self-crossing. The progenies of such a cross can be easily be analyzed to predict the genotype of the test organisms. Figure 4.5 shows the results of typical test cross where violet color flower V is dominant over white color flower small v. Using Punnett square, try to find out the nature of offsprings of a test cross. What ratio did you get? Using the genotype of this cross, can you give a general definition for a test cross? Based on his observations on monohybrid crosses, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called the principles or laws of inheritance. The first law or law of dominance and the second law of segregation.